Hello and welcome to another lesson. In today's lesson, we'll be learning about enzymes, specifically digestive enzymes. At the end of this video, you should know the types of enzymes, the classification of enzymes, their characteristics, their functions, and you should also know some examples of enzymes. An enzyme is a biological molecule, usually proteins, that act as catalysts in living cells. They speed up chemical reactions without being used up in the process. This is possible because they act by lowering the activation energy of reactions. Think of them as tools used to make things run efficiently. Digestive enzymes can be found in the mouth, the stomach, and the small intestine. So, enzymes have some unique characteristics. Let's take a look at some of them. Specificity. Enzymes are highly specific in nature. That means they only act on one type of molecule. For example, lactase only works on lactose. Another characteristic is optimal conditions. That means they work best under specific temperatures and pH levels. For example, pepsin only works in acidic environments of the stomach, and trypsin needs the slightly alkaline pH in the small intestine. So, pepsin needs an acidic pH, and trypsin needs an alkaline pH to work. Another characteristic of enzymes is reusability. Enzymes don't get used up in reactions. They are like reusable tools. They make the end product without changing its chemical composition, and that means they are not used up in the process and are ready for another round of work. Sensitivity is also a very important characteristic of enzymes. So if the conditions are too harsh, enzymes can be denatured or deactivated. And this can be seen in conditions like extreme heat or pH changes. And that's one of the reasons why your body needs to always be in the right conditions. We're talking right temperature, right pressure, and stuff like that. If you're still watching, hopefully you found this video helpful. Please support the channel by tapping the thumbs up button, the subscribe button, and the share button. If there's something you don't understand, you can ask for clarification in the comments. So let's look at factors that affect enzyme activity. We have temperature, pH levels, substrate concentration, and inhibitors. So, temperature. Optimal temperature increases activity, and extreme temperature denatures the enzymes. pH levels. Each enzyme works best at a specific pH range. We have acids and then we have alkalines. So, enzymes that require alkaline mediums must have alkaline mediums to work. So, substrate concentration. Higher substrate levels will increase the activity of enzymes. And then finally, inhibitors. Inhibitors are molecules that interact with enzymes. It can either be permanent or temporary, and either reduce the rate of an enzyme catalyzed reaction or prevent enzymes from working completely. So, why exactly is it important for us to learn about enzymes, and why are enzymes actually important? Why doesn't the body just, convert our food on its own? So enzymes help in quickly breaking down the large molecules of ingested food like starch, proteins, and fats and oils into tiny molecules the cell can easily absorb. Without enzymes, cells would starve before nutrients reach them. So that means the time taken for the body to normally convert these large molecules will be much, much longer. And most of the cells require constant supply of energy to function properly. So, what are the types of enzymes? The enzyme types depend on the substrates that they act on. So we have carbohydrate digesting enzymes, protein digesting enzymes, fat digesting enzymes, and nucleic acid digesting enzymes. Under carbohydrate digesting enzymes, we have amylase, which breaks down starch into maltose and glucose. When found in the saliva, it is called salivary amylase. When found in the pancreas, it is called pancreatic amylase. We also have maltase, which converts maltose to glucose. We have lactase, which breaks down lactose, that is milk sugar, into glucose and galactose. Finally, we have sucrase, which converts sucrose, that means your regular table sugar, into glucose and fructose. Under protein digesting enzymes, we have pepsin, which breaks proteins into smaller peptides in the stomach. We have trypsin and chymotrypsin, which are produced in the pancreas, and these enzymes further break down peptides into smaller chains in the small intestines. We also have peptidases, which convert peptides into individual amino acids. Under fat digesting enzymes, we have lipase. And lipase breaks down fats into glycerol and free fatty acids. Lipase is secreted by the pancreas and works in the small intestine. We also have bile, and although bile is not an enzyme, bile from the liver emulsifies fats and makes them easier for lipase to digest. And then finally, we have nucleic acid digesting enzymes. Under nucleic acid digesting enzymes, we have nucleases, and these are responsible for breaking down DNA and RNA into nucleotides. Examples include deoxyribonuclease, which is DNase, and ribonuclease, which is RNase. All the aforementioned enzymes are either intracellular enzymes or extracellular enzymes. Intracellular enzymes work inside the cells of the living organisms, and extracellular enzymes carry out their functions outside the cell. Examples of extracellular enzymes are digestive enzymes. So let's move on to the classification of enzymes. Enzymes are grouped into six major classes based on the types of reactions they catalyze. Let's go through them one by one, starting with oxidoreductases. Oxidoreductases are in charge of oxidation reduction reactions. This means they help move electrons from one molecule to another. They are involved in reactions like oxidation of alcohols and the reduction of oxygen in respiration. Under this group, we can find enzymes like the hydrogenases, which catalyze the removal of hydrogen atoms, oxidases, which add oxygen to substrates, and peroxidases that break down peroxides. These enzymes all have their own specific example. Take lactate dehydrogenase, for example. It helps convert lactate to pyruvate by removing hydrogen atoms. So if you're breathing oxygen and producing energy, you have oxidoreductases to thank. Next, transferases. These enzymes are like delivery drivers, transferring functional groups like a phosphate or a methyl group from one molecule to another. We can find enzymes like kinases and transaminases in this group. Kinases transfer a phosphate group from a high energy molecule, typically ATP, to a substrate, and transaminases transfer amino groups. For a specific example, we have hexokinase, and it's a kinase that adds a phosphate to glucose in glycolysis. So moving on to number three, we have hydrolases. Hydrolases are known as the bond breakers. These enzymes use water to break down molecules. They're responsible for breaking of peptide bonds, ester bonds, and glycosidic bonds. We can find enzymes like proteases, lipases, and amylases in this group. So proteases break down proteins, lipases break down fats, and amylases break down starch. For a specific example, we have trypsin, which is a protease that helps to digest protein. Liases are up next. They catalyze the addition or removal of groups without hydrolysis, often forming double bonds or rings. They're responsible for things like removal of water or carbon dioxide from substrates. We can find enzymes like decarboxylases, synthases, and hydrolases in this group. Decarboxylases remove carboxyl groups, synthases facilitate addition reactions, and hydrolases catalyze the breakage of a carbon-oxygen bond, leading to unsaturated produce via the removal of water. For a specific example, we have fumarase, which is a hydrolase used in the citric acid cycle to remove a molecule of water to form fumarate. Isomerases. Have you ever rearranged your furniture to make your room look better? That is what isomerases do, but with molecules. They catalyze the rearrangement of atoms within a molecule to form isomers. We can find enzymes like epimerases and mutases in this group. Epimerases change stereochemistry at one carbon. Mutases transfer functional groups within a molecule. Last but not least, we have ligases. These are the glue guns in the enzyme world. They join two molecules together, often using ATP. DNA ligase is a classic example. It seals breaks in DNA strands, keeping the genetic code intact. So, we have covered the six classes. To help you remember, here's a mnemonic. On the hot lips I linger. O represents oxidoreductases, T stands for transferases, H for hydrolases, L for lyases, I for isomerases, and L for ligases. Easy, right? From this classification, we can easily see that all digestive enzymes belong to the class called hydrolases. The hydrolase class generally refers to enzymes that break up various bonds. That means breaking large molecules into smaller molecules in the presence of water. So that's it for the video. Were you able to watch the video to the end? Drop a smiley face in the comments if you made it to the end of the video. Don't forget to show your support by subscribing to the channel, liking and sharing this video.